Hello, this is me IramGul and you are watching Chem Tracks by IGM. As you know, in our previous lectures we have done detailed discussion on carbohydrate and proteins. Today we are going to start another biomolecule, that is lipids. Lipids are one among the four major biomolecules of living systems. By definition, these are the class of biomolecules which are insoluble or sparingly soluble in the aqueous solutions and soluble in organic solvents. Fatty acid are major constituents of lipids. Fatty acid are monocarboxylic acid containing short and long chain hydrocarbon molecules. The numbering of carbon in fatty acid begins with the number with the carbon of the carboxylate group. Fatty acid represented by the total number of carbon, that is palmitic acid, a 16 carbon fatty acid, and its formula is CH3CH214COOH. This is basically carboxylate group, and it is designated as a C16, mean it contain uh, 16 carbon atom in chain. It is customary to write as C ratio 16 ratio 0, where 0 represents that there is no double bond in the fatty acid. If there is one double bond, then it will be written as C ratio 16 ratio 1. These are some common fatty acid. First is butyric acid, and it is represented by 4 ratio 0, mean it contains 4 carbons and 0 double bond or no double bond. And its formula is CH3CH2 twice COOH. Caproic acid is a six-member chain with no double bond, and its formula is CH3CH2 4COOH. Decanoic acid, as name indicated, deca mean 10, so it contains 10 carbon atoms. Lauric acid is 16 member fatty acid. Myristic acid is a 14-member fatty acid, CH3CH2 12COOH. Palmitic acid, as we discussed earlier, it is 16 carbon member fatty acid. Stearic acid is a 18 carbon fatty acid, CH3CH2 16COOH. Arachidic acid is another example of common fatty acids, and consists of 20 carbons with no double bond. Its formula is CH3CH2 18COOH. 18 mean there are basically total 18 carbons are here, and this is the 91th and 20th carbon. On the basis of double bonds, there are two types of fatty acid: saturated fatty acid and unsaturated fatty acid. All sets of example in the previous table were fatty acid that contained no carbon-carbon double bonds. These are called saturated fatty acids. Just we discussed diuric, caproic acid, decanoic acid, lauric acid, myristic acid, palmitic acid, stearic acid, and arachidic acid. These are the common example of saturated fatty acid. Saturated fatty acid having a short carbon chain are liquid at room temperature, whereas the long chain carbon fatty acids are solid at room temperature. Another type of fatty acid is unsaturated fatty acid. These have carbon-carbon double bonds in between, thus leading to unsaturation. This is very important. The representation for these fatty acids consists of number of carbon atoms followed by the number of double bond in the place of unsaturation. So we have to consider three things. First, the total number of carbon atoms. Second, the total number of double bonds. And the third, the place of unsaturation or the place of double bond. The place of unsaturation in fatty acid is indicated by the symbol delta, and the number of first carbon of the double bond is superscript form. There are basically three common nomenclature used to represent the fatty acid: omega, delta, and common are trivial names. So here we are using delta nomenclature for representation of unsaturated fatty acid. Thus, oleic acid is a 16 carbon fatty acid with one site of unsaturation or with one double bond between carbon 9 and 12. So, it will be represented by C16, mean there are 16 carbon, ratio 1 delta 9. 1 delta mean one double bond, and 9 mean it is double bond is present as at position number 9. So, there are some common oleic acid just we discussed here. Linoleic acid is a 18-member fatty acid. It consists, It contains two double bond, two delta 9 and 12. Two delta mean two double bonds, 9 and 12 are position of these two double bonds. So you can see there are two double bond in its formula. Linolenic acid is another example of unsaturated fatty acid. It also based on 18 carbons and 3 double bonds at position number 9, 12, and 15. Arachidonic acid is um, 20 carbon fatty acid consist of four double bonds at position number 5, 8, 11, and 4. Lipids are biologically important. Lipids perform or involved in variety of important cellular functions. However, following are the some major physiological functions attributed to lipids. Energy source in animals, insect, birds, and high lipid seeds that are triacylglycerol. Activators of enzymes, namely glucose 6 phosphatase, steroyl CoA desaturase, monooxygenases, which are important mitochondrial enzymes. Some of the lipids derivatives serve as vitamins and hormones that are prostaglandins. Arachidonic acid is the specific precursor for all prostaglandins and leukotrienes. It is bound as an acyl moiety to the two position of a number of phospholipids, and this is inactive as a substrate. Upon release by the action of phospholipase A2, the free arachidonic acid is converted via cyclooxygenase to form two very prostaglandins derivative, prostacyclin 12, an active physiological vasodilator, and thromboxane A2, an active vasoconstrictor. Essential components of biological membranes that are sphingolipids and glycolipids. They are used as a lipoproteins in protein modification and recognitions. They are components of the electron transport system in the inner membrane of mitochondria. This is basically a short or brief introduction about the lipids. In our coming lectures, we will discuss it in details. Keep watching and goodbye.